Good morning, students. Today we will discuss about the relationship between gene and enzyme. Now, what is gene? The term gene was introduced by Johannesson in 1909. Prior to him, Mendel has used the word factor for a specific, distinct, particulate unit of inheritance that takes part in expression of a trait. So, Johannesson has defined gene as an elementary unit of inheritance which can be assigned to a particular trait. Okay. Now, presently, a gene is defined as a unit of inheritance composed of a segment of DNA or chromosome situated at a specific locus which carries coded information associated with a specific function and can undergo crossing over as well as mutation. So we can say that gene is a unit of hereditary information that occupies a fixed position on a chromosome. Here you can see. Okay, so they are a distinct sequence of nucleotide that form a part of the chromosome, and gene are found present in almost every single cell's nucleus and are composed of DNA strand. So it is constituted by the segment, okay, wherein each gene adds a particular protein to the final outcome. Now, modern concept of gene. Gene as a unit of transmission of cistron. That means the part of the DNA specifying a single polypeptide chain is called a cistron. Okay, so a cistron is a segment of DNA that specify a particular function. Next, it is a unit of recombination and or recon. That means gene is a specific region of the chromosome determining a distinct trait in the organism. Okay, a small segment of DNA capable of being separated and exchanged with other chromosome is called a recon. And then it is a unit of mutation or muton. That means the smallest unit of genetic material which when changed or mutated produces a phenotypic trait. Okay, means study of mutation shows that a change in a trait may result from an alteration in the structure of a chromosome or a sequence of bases or even a single base. Thus, a gene may be defined as a shortest segment of a chromosome which can undergo mutation. Okay. Now, what are the characteristics of genes? So, genes are the fundamental unit of hereditary variation, mutation and evolution. So, genes determine the physical as well as the physiological characteristic of organism. Okay, and they are responsible for transferring these characters from parents to the offspring. They are situated on chromosome and they occupies a fixed position which is called a locus. They are arranged in single linear order in a chromosome as beads on a string, and they are capable of replication. That means they are able to replicate, and it is a unit of recombination, so they are capable of undergoing crossing over, and it is a unit of genetic material which undergoes mutation. Okay, so it can undergo mutation to form an allele with a different expression. Okay, and somatic structure. That means it is a unit of heredity connected with somatic structure of functions that leads to a phenotypic expression. Okay, so thus gene regulates structure or metabolism. Okay, now next is genes and enzyme. Now, the father of the biochemical genetics is Sir Archibald Garrod. Okay, he initiated the analysis of inborn errors of metabolism in humans in terms of biochemical genetics. Okay, means the most important characteristic of the gene is to store and express the genetic information that will contribute towards the phenotype and will passed on to successive generation. So how this occurs? So the idea that gene controls the metabolism was put forward as early as 1902 by Garrod. So he studied a number of several disorders which seem to be inherited. Okay, and he found that they are the inborn errors of metabolism or failure of metabolic machinery of the organism to perform a particular function due to formation of some defective enzyme associated with inheritance of defective genes. Okay, and hence he called them the inborn error of metabolism. So in one such disorder, that is Alkaptonuria, affected individual cannot metabolize, okay, alkapton. Okay, now this alkapton is produced in human being, and this is formed during metabolism of two amino acid. Okay, this is phenylalanine and tyrosine. Okay, and normally alkapton is oxidized to carbon dioxide and water with the help of alkapton oxidase. Okay, so in the patient suffering from alkaptonuria, the enzyme is defective and unable to metabolize the acid, and as a result this alkapton is accumulated. Okay, so it gets accumulated and then it is excreted in the urine, and the oxidation product of this molecule are black, and thus it can be easily detected by the blackening of the urine exposure. Okay, when it is exposed to the air. Okay, so Garrod found that this disorder is inherited as a recessive trait. Okay, and reason that normal individual must be metabolizing alkapton. Okay, so and in affected individual this metabolic pathway is blocked, which results into the disorder that is alkaptonuria. Okay, so he studied this defect, that is a genetic disorder or the disease of the human beings, which is characterized by brown or black color of the exposed unit. Okay, and he came to the conclusion through pedigree analysis that the disease was caused by the inheritance of a pair of recessive gene. Okay, so a number of the human diseases were identified and described which fell into the category of inherited metabolic defects. Okay, and Garrod referred to this as an inborn error of metabolism. Okay, and he found other diseases that followed similar patterns. So it thus led to the idea that many disorder may be inherited as any other gene, that genes function by controlling the metabolism.